Assalamu alaikum. My name is Hamna Shams from the department BSBT. My today's topic of presentation is ELISA reader. So what is ELISA reader basically? Introduction to ELISA reader. Enzyme linked immunosorbent assay is a labeled immunoassay that is considered the gold standard of immunoassays. This immunological test is very sensitive and is used to detect and quantify substances including antibodies, antigens, proteins, glycoproteins, and hormones. The detection of these products is accomplished by complexing antibodies and antigens to produce a measurable result. An antibody is a type of protein produced by an individual's immune system. This protein type has specific regions that bind to antigen. An antigen is a protein that can come from foreign source and when bound to an antibody induces a cascade of events through the body's immune system. This interaction is utilized in ELISA testing and allows for identifying specific protein, antibodies and antigens with only small amount of a test sample. Importance in biochemical assays. ELISA testing is used to diagnose HIV infection, pregnancy test and blood typing among others. The design of an ELISA reader is typically very simple. The reader typically consists of a plate with a set of wells, each well containing a different antigen. The reader is then used to detect the presence of the antigen in a sample. Components. An ELISA reader typically includes a plate with wells, a detector, and a sample. The plate has wells that are designed to hold the sample, and the detector is used to detect the presence of a specific antigen or antibody in the sample. The first component, light source. An ELISA reader typically includes a light source to illuminate the plate and detector to detect the presence of a specific antigen or antibody in the sample. Filters. The second component. Filters can be used to remove certain substances from sample before it is analyzed. Microplate handlers, the third one. Microplate handlers are used to transfer samples from one location to another. The fourth one is photodetectors. Photodetectors are used to detect light and convert it into an electrical signals. These signals can then be used to identify objects or substances. Picture of ELISA assay. Now manufacturing of ELISA reader. Fabrication of microplates. The fabrication of microplates involves the production of raw materials, the assembly of the microplate, and the packaging. In ELISA, the raw materials are the antigen and the antibody. The assembly of the microplates involves the addition of the antigen to the wells of the microplate and the addition of the antibody to the wells of the microplate. The packaging of the microplates involves the sealing of the wells of the microplate with the adhesive. Assembly and quality control. The quality control of the assembly of the microplates involves the testing of the microplates for the presence of the antigen and the antibody. This is done to ensure that the microplates are correctly assembled and that the antigen and antibody are present in the correct concentrations. Principle and working of ELISA. The principle behind the ELISA assay is to detect the presence of a specific antigen or antibody in a sample. This is done by incubating the sample with a specific antibody or antigen and then measuring the amount of antigen or antibody that is present. Now the working of ELISA. The ELISA assay works by incubating a sample with a specific antibody or antigen. This will cause the antibody or antigen to bind to the sample and then the amount of antibody or antigen present can be measured. Now types of ELISA assays. There are four major types of ELISA. Direct ELISA, indirect ELISA, sandwich ELISA and competitive ELISA. There are four main general steps to completing an ELISA immunoassay. These steps are: number 1, coating with either antigen or antibody. Number 2, blocking, typically with the addition of bovine serum albumin. Number 3, detection, and the last is final reader. Detection. Detection is carried out by the addition of a substrate that can generate a color. There are many substrates available for use in ELISA detection. However, the most commonly used horseradish, peroxidase, and alkaline phosphatase. The substrate for HRP is hydrogen peroxide and results in a blue color change. ALP measures the yellow color of nitrophenol after room temperature incubation periods of 15 to 30 minutes and usually use p-nitrophenyl phosphate as its substrate. Between each of the above four steps is a wash of the plate using a buffer such as phosphate buffer saline and a non-ionic detergent to remove unbound material. The wells are washed 2 or more times during each wash step, depending on the specific protocol being followed. In the ELISA protocol, usually a serial dilution of concentration is placed in the wells of the plate. After the results are measured, a standard curve from the serial dilutions data is plotted with the concentration on the x-axis using a long scale and absorbance on the y-axis using a linear scale. Now the types. The first type is direct ELISA. Both direct and indirect ELISA begin with the coating of antigen to the ELISA plates. The first binding step involves adding antigen to the plates, which is incubated for 1 hour at 37 degrees Celsius or can be incubated at 4 degrees Celsius overnight. Once the incubation step is completed, the next step is to wash the plates of any potential unbound antibody and block any unbound sites on the ELISA plate using agents like BSA, ovalbumin, aprotenin, or other animal proteins. This second step is important because it prevents the binding of any non-specific antibodies to the plate and minimizing false positive results. After adding the buffer, the plate is re-washed and a selected enzyme conjugated primary detection antibody is added. The plate is further incubated for 1 hour. In a direct ELISA, the primary detection antibody binds directly to the protein of interest. Next, the plate is re-washed to remove any unbound antibody and followed by the addition of substrate chromophore such as alkaline phosphatase or horseradish peroxidase to the plate, which results in a color change. The color change is a sample occurs by either the hydrolysis of phosphate groups from the substrate by AP or by the oxidation of substrate by HRP. The advantages of using direct ELISA include eliminating secondary antibody cross-reactivity, and due to fewer steps, it is rapid compared to indirect ELISA. Its disadvantages include its low sensitivity compared to the other types of ELISA and its high cost of reaction. Direct ELISA. Indirect ELISA. The steps of the indirect ELISA are identical to the direct ELISA, except for an additional wash step and the types of antibody added after the buffer is removed. Indirect ELISA requires two antibodies, a primary detection antibody that sticks to the protein of interest and secondary enzyme-linked antibody complementary to the primary antibody. The primary antibody is added first, followed by a wash step, and then the enzyme conjugated secondary antibody is added and incubated. After this, the steps are the same as the direct ELISA, which includes a wash step, the addition of a substrate, and the detection of a color change. The indirect ELISA has a higher sensitivity when compared to the direct ELISA. It is also less expensive and more flexible due to many possible primary antibodies that can be used. The only major disadvantage with this type of ELISA is a risk of cross-reactivity between the secondary detection antibodies. Indirect ELISA.